Hello everyone, my name is Đurađ Milošević and I'm from the University of Niš, Serbia. I'm gonna talk about artificial intelligence in species monitoring as a powerful tool which can automate the whole process. This study is just a part of the big project where we want to develop the application which can identify species using image recognition approach. So the challenge is to improve the efficiency of species monitoring, which can consequently increase the cost effectiveness of all studies which use species monitoring, such as biomonitoring programs and biodiversity monitoring. And to tackle this challenge, we want to do it through interdisciplinarity, and we gathered the interdisciplinary team, which involves not only biologists and taxonomists, but also IT experts, because we wanted to apply sophisticated IT methods. However, let's talk about the problem regarding this challenge. So we found the main problem in the routine monitoring programs, the identification of species, because this process is slow, it's really time consuming, and it almost always requires expertise, which is unfortunately missing, especially for some species groups, such as chironomids. We call this group dark taxa in biomonitoring because they have really complicating identification, unknown taxonomy for some species, and this process always requires expertise. For example, look at these species which are presenting from the lateral point of view. They are almost similar, especially for non-experts. Because of that, chironomids, beside the huge potential in as indicators in bioassessment, they're excluded from almost all routine monitoring programs. So we wanted to change this, and we decided to call machines for help. Of course, we called IT experts, and we wanted to use artificial intelligence to sort out this problem. To do that, we applied simultaneously 3 different approaches in identification. First, we used the traditional one based on morphology characteristics. Then we use a DNA approach to validate these different morphology hypotheses. And finally, we use machine learning approach, which is based on computer vision and deep learning. So it is planned to first identify all species using morphology-based approach, then to validate this identification using DNA approach, and then to photograph all species and to make a huge image data set. First, we tried with only 10 species, including 250 specimens per each species. So we photograph all of them and use this image data set to train our neural network. For that purpose, we use convolutional neural network, which is the best type of neural networks for that task, image classification. So briefly, the composition of neural network is the following. We can recognize three different layers here, type of layers here. The leftmost layer is the input layer, which is exposed to raw data, in our case, chironomid images. The rightmost layer is the output layer, and with hidden layers in between. There are usually from 5 to 100 layers for deep learning, which we use here. So how it goes? As data, after we introduce image data set into the network, and as data moves from the lowest layer to the highest layer, more abstracted information is collected at increasing scale from small edges, points, to object parts, and eventually the entire objects. For example, in our case, mouth parts, such as mandibles, mentum, and so on. When trained, the network forms a hierarchy of image structures, image features, with increasing complexity, starting from low-level image concepts. As I said, this considers just small dots or edges, lines, to the high-level image concepts, which are close to the output layer, including parts of the object and the whole objects. So after the training process, the network will be capable to identify and to classify species by itself without any expertise. And we did this. We made a network with extremely high level of accuracy, higher than 99.5%, which is fabulous. And we also visualize the inside of the network and reveal which features the network used for decision making, for classification. And what was really interesting is that these features mainly overlap with the one used in identification keys. However, the whole this approach, convolutional neural network, is supervised approach, meaning that we introduce a labeled specimen into the network. So network, during the training process, network knows to which species each specimen belongs. However, we recognize that as a semi-automatic approach, and we wanted to go one step further and to apply unsupervised neural network, which can process non-labeled entities. So the objective was to apply a deep learning based approach to test the efficiency of an unsupervised algorithm in automatic classification of species of chironomids. So we use the same data set, but this time completely different architecture of neural network. For this purpose, we use autoencoder. Autoencoders are an example of a deep neural network working in an unsupervised manner. So the inputs are used as learning examples, and the outputs are scored based on their similarity to the inputs. The autoencoders are simple networks that attempt to reconstruct inputs with a little noise as possible. So the input is, in our case, image data set. Using encoder, it will be transformed into the vector. And after that, the decoder will reconstruct from the vector the image with a minimum information loss. So this is a huge set of different autoencoders, and for our study we used beta variation autoencoders, which can learn explicit representations that are faithful to data generative factors, which is known as a learning of disentangled representations. It includes single latent encoding units which correspond to individual and independent generative factor in the image. So imagine the image, one image of chironomid as a highly dimensional set of information. So a key idea of disentangle representation learning is that while the data sample from the real world appears to be extremely high dimensional, a small number of latent or hidden factors are responsible for their variation. So beta variation autoencoders can recognize these representations and present them via vectors. So the output which we use here is actually transform photo into the vector with the most important features. And then these vectors, the variability of these vectors was visualized using one of the best dimensional reduction approach, which is called uniform manifold approximation and projection. Imagine this approach as a I would say substantially improved principal component analysis. However, it deploys manifold learning, which is capable to identify clusters in the data set while constraining the size of the local neighborhood. So this approach can retain both local and global relationship between entities in the data set. The UMAP approach can visualize the variability within data set and visualize and reveal the similarity between these entities. And let's see the results when we use beta variational autoencoder on our data set, which is composed of 10 species and 250 specimens per species. So what we can see here is the ordination pattern of each specimen based on these most important species, most important features abstracted by beta variational autoencoder. What we can see here, each specimen is coded by color based on previous identification, based on morphological features and traditional identification approach. So what we can see here is that betaVAE algorithm separated the same specimens from the same species, Chironomid riparius, And probably since this species is composed of usually two different species, chironomid riparius and piger, probably we made a mistake during identification, and betaVAE algorithm recognized that mistake and made two different species. Also, what was interesting is that betaVAE algorithm couldn't make a difference between Tvetania species, because we introduced here Tvetania discolor repeat aggregate and Tvetania clavescens. So the algorithm put them together and didn't make a difference. The second interesting thing was the similarity recognized by betaVAE between two completely different species from different genera, Chironomus nudiventris and Polypedilum nubeculosum. What was similar between these two species is the submentum region, which is colored in both species. From this ordinational pattern, we can see that betaVAE algorithm put also close together two different species, Microtendipes pedellus and Polypedilum scalenum, probably because of medium teeth, actually probably because of the mentum, which has similar pattern of teeth. And also betaVAE algorithm and UMAP approach put close together three different species from the same genus, Polypedilum, and Polypedilum scalenum, Polypedilum latum, and convictum. And this is something what was expected because these three species were really similar. So we can conclude that betaVAE algorithm and computer vision approach avoids limitations in taxonomic knowledge by automating the identification of specimens without any supervision, and that betaVAE approach without any supervision classify chironomids in particular taxonomic units. And this classification was fully consistent with the supervised deep learning approach, which we conducted previously before the study. So using deep learning approaches, both actually supervised and unsupervised, could be possible to substantially increase the reliability and the use of chironomids in bioassessment programs. So thank you for your attention. And I just wanted to say that this research was supported by the Science Fund of the Republic of Serbia. If you are interested in the whole project, you can find our Twitter account. So one more time, thank you for your attention.